All it aspirants, you are listening to podcast Botany furnished by S. Ramakrishna. In this podcast, the types of RNA and protein synthesis. RNAs in prokaryote as well as eukaryote exhibit division of labor. There are different types of non-genetic RNA, mRNA, rRNA, tRNA. mRNA carries the genetic message from the DNA. Now this mRNA having genetic message for particular protein, polypeptide. So mRNA is a template for polypeptide. rRNA present in ribosome, it is a structural as well as catalytic. So all catalytic are structural RNA, but all structural RNA are not a catalytic. Some reactions catalyzed by nucleic acid enzyme, that is catalytic RNA, ribozyme. tRNA, transfer RNA, otherwise it is called adapter RNA. It is interpreter of genetic code. It reads the genetic code present on mRNA and brings the amino acid. Next. So all these three types of RNA transcribed by one RNA polymerase in prokaryote. So there is no division of labor for RNA polymerase in prokaryote, but there is a division of labor for RNA, mRNA, rRNA, tRNA in protein synthesis. So DNA dependent RNA polymerase ribonucleoside triphosphate in template dependent fashion, obeying the rule of what it is complementarity. Here, what are the raw material or substrate for transcription in this process? Ribonucleoside triphosphate. What are the raw materials in case of DNA replication? Deoxyribonucleoside triphosphate. Next. The RNA polymerase binds to promoter and initiate transcription, and a short stretch of RNA attached to RNA polymerase. Once RNA polymerase reached to terminator, the nascent RNA as well as RNA polymerase falls off. An intriguing question, a mind-blowing question is how RNA polymerase catalyzing all these three reactions in case of transcription, that is initiation, elongation, termination. Because RNA polymerase alone can do only elongation, somehow responsible for opening of DNA helix during transcription. Here also one question. Which enzyme involved in opening of helix in transcription? RNA polymerase. Which enzyme involved in opening of DNA helix in DNA replication? That is helicase. Here RNA polymerase. The answer it is RNA polymerase changes its specificity. Its specificity is elongation, but changes its specificity by transiently, temporarily associating with a protein factor sigma and other factor, protein factor rho. So RNA polymerase once associated with the sigma initiate transcription. When it associates with the rho factor, it terminate transcription. So for initiation sigma factor, for termination it is rho factor. Next. So transcription in this initiation sigma. RNA polymerase can alone do elongation. With the rho factor, termination. Next. In bacteria, transcription and translation are coupled. That means transcription, translation occur in a one compartment, single compartment, because there is no demarcation of cytoplasm and nucleoplasm. Why? There is no nuclear membrane. The second one is RNA need not undergo any processing to become active. Why? Genes are non-split in case of prokaryote. Third, in many cases, translation begin much before mRNA fully transcribed. Once the transcription initiated means simultaneously it is almost Transcription, translation are coupled. Suppose imagine in case of mRNA A, there is no amino acid brought by tRNA. U, there is no amino acid brought by tRNA. AUG on mRNA. tRNA brings the amino acid. So, transcription, translation almost simultaneous. That's why transcription, translation are coupled in case of bacteria. In eukaryote, there is a complexity with regard to RNA polymerase. The first complexity, RNA polymerase exhibit division of labor in contrast to the prokaryote. That is, many types of RNA polymerase are present. There are at least 3 RNA polymerases in nucleus in addition to organelle RNA polymerase. So how can you say the minimum number of RNA polymerases in case of chlorella? Chlorella, it is an algae. It is having mitochondria as well as chloroplast. Nucleic acids are present in chloroplast and mitochondria. So in nucleus, 3 RNA polymerases, chloroplast minimum 1 and mitochondria 1. So 5 minimum RNA polymerases are expected in case of green alga chlorella. What about the fungi? Suppose if we take rhizopus, no chloroplast. So in nucleus, 3 types of RNA polymerases, and in mitochondria, 1 minimum type of RNA polymerase. So 4. So there are 3 RNA polymerase at least in case of the nucleus in addition to RNA polymerases. Next, RNA polymerase 1, which transcribe 5.8S rRNA and 18S rRNA, 28S rRNA. S indicates sedimentation coefficient. RNA polymerase 2 transcribe the precursor of mRNA. Precursor of mRNA is hnRNA, heterogeneous RNA. It is having exons as well as intron. RNA polymerase 3, which transcribe 5S RNA, tRNA, snRNA, small nuclear ribonucleic acid, which involved in post-transcriptional gene regulation, that is splicing process. Next, it is the first complexity. The additional complexity is just formed RNA transcript is non-functional because it is having exons as well as introns. Exons are expressive sequences, introns which are not expressive sequences. So it should undergo a processing. Intron should be removed, exon should be joined. So introns are removed by spliceosome, that is snRNA along with ribosome, which forms spliceosome, which remove this intron in a looped form. So intronic loops are formed which are removed. Later the exons are joined by RNA ligase. That is one thing. And there is an additional processing as this RNA has to leave the nucleus, enter into cytoplasm. It needs protection from other enzyme to prevent from being cleaved. So a processing called capping and tailing. Just after transcription initiated, capping, that is capping is nothing but an unusual nucleotide. Why you are calling it the unusual nucleotide? 7-methyl guanosine triphosphate. Guanine actually it is 2-amino-6-oxypurine. On 7th carbon, there is a methyl group added. So 7-methyl guanosine triphosphate added to hnRNA in template independent fashion. Capping. This cap protect RNA from being degraded or it is enzymatic action from other enzyme, and also helps to recognize the ribosome during translation. That is the advantage. Next, tailing. 200 to 300 adenylate residues are added at the 3 end of hnRNA in a template independent fashion. So capping and tailing. So capping prevent RNA from being cleaved from 5 end. Tailing prevent RNA from being cleaved from 3 end. So RNA got protection. Now processed hnRNA that is present in case of nucleus, now we are calling it as mRNA, leaves the nucleus and enter into cytoplasm, going to associate with the ribosome for protein synthesis. So mature RNA does not have introns, only it is having exonic sequences. Next. Presence of introns is the reminiscent of antiquity. The antiquity is the old character, the primitive character. Reminiscent of antiquity. So initial genetic material it was RNA, RNA having introns and exon, and DNA evolved from RNA having both introns and exons. Now the eukaryote showing introns and exon. So it is supposed to be evolved from RNA. So presence of introns is reminiscent of antiquity. Next. Split gene arrangement is the ancient feature of genome. In earlier genome, the gene it was fragmented, that is exons and introns present. As DNA evolved from this, it is also having introns and exon. So split gene arrangement is the ancient feature of genome. Third, splicing shows the dominance of RNA because DNA is a static storage of exons and introns. RNA can process and regulate to kept which introns, to delete, to kept which exons, to delete which intron, and when protein has to be formed, at which stage the protein has to be formed, decided with the splicing, that is alternate splicing. So DNA is not going to decide anything. RNA can regulate the expression with the help of, that is splicing, that is alternative splicing. Suppose exon 1, 2, 3, 4. Exon 1, 2, 3, 4 joined for one protein. Exon 1, 4, 7, 8 are joined for one type of protein. So there is a great genetic diversity, different types of isoprotein formed from just few genes. So that is the advantage in case of eukaryote. I am signing off from this episode. We will see in case of the next episode, the tRNA and genetic code.